Hey guys, today we're going to be talking about cell walls and biosorption. I know you might be thinking this sounds like a boring topic, or maybe you're not because you clicked on this video and that is the title. But I hope you're interested because this is a majorly important topic just to understand before I can move on to some really cool and interesting topics on my channel like geobiology and astrobiology. And yes, astrobiology means biology in space, guys, and understanding a little bit about some background in biology and geobiology is important to understand astrobiology because the intersection where biology and geology meet is really, really cool. Now, this lecture specifically is also really important for something called bioremediation and biorecovery, and I'll talk about those in a separate video after we talk about the basics behind biosorption in this video. So let's get started. Before we can talk about biosorption itself and how it can help us with biorecovery and bioremediation, we need to talk about cell walls. So for the outline of this video, we'll be talking about gram-positive versus gram-negative bacterial cell walls and how they're structured, because how they're structured really does affect how they absorb materials, and bacterial surface layers, because the cell wall may not be the most outer layer of the bacterium or the group of bacteria in a particular area. They may be excreting some material called EPS, sheaths, or they may have S layers. And we'll talk about all three of those types of surface layers for bacterial cells as well. Then we'll move on to archaea and their cell wall structures, and then eukaryotes, and talk a little bit about how plant, animal, and fungal and algal cell walls differ from that of prokaryotes. Then we can move on to biosorption and talk a little bit about microbial surface charge and how passive biosorption can happen because of the charge of microbial surfaces, and also a little bit about active absorption and what the difference between passive and active biosorption is. So let's get into it. First, bacterial cell walls. Recall from previous videos I've done that bacteria have cell walls which encapsulate their cytoplasm, which within the cytoplasm is their genetic material. So bacteria and prokaryotes in general are pretty simple. They've got their cytoplasm, or their internal cell material, in which they have their genetic material, and surrounding that they've got their cell wall. They don't have all of the crazy organelles that eukaryotes have, as shown here to the right. And I give the example of mitochondria, well, for two reasons. One, pretty much everybody knows mitochondria as the powerhouse of the cell. That's commonly known in biology lingo and intro classes, and it is, but that's for eukaryote cells. Prokaryote cells don't have mitochondria. Instead, all of the powerhouse activity that happens in the mitochondria of eukaryotes happens in the cell wall, or really just the plasma membrane of prokaryotic cells. And when I say prokaryotic, I mean bacteria and archaea. Those are the two main groups of prokaryotes that we'll be talking about. And the first one we'll be talking about is bacteria. Bacteria have two main cell wall types. These types are called gram-positive and gram-negative. This is because they're differentiated based on a staining technique in which we stain them using something called gram stain, which is a series of chemicals we put onto the bacterium and see if it retains a purple or a pink color. If it retains a purple color, it is gram-positive. If it retains a pink color, it is gram-negative. Gram-positive cell walls can be seen up here to the top right, and gram-negative to the bottom right. Gram-positive, as we can see, have a large purple area, which is called their peptidoglycan, which is just a network of linear polysaccharide strands connected by proteins. But basically, just remember that the peptidoglycan is really thick in gram-positive cells, whereas in gram-negative cell walls, it's relatively thinner, and it's surrounded in gram-negative cell walls by two membranes, whereas in gram-positive, there's only the inner plasma membrane. So in gram-negative cells, we see there's this outermost membrane structure that is aptly named the outer membrane. And so that's the main difference between gram-positive and gram-negative bacterial cell walls. So basically, the gram-positive essentially contain two parts, just to sum it up, the membrane and the peptidoglycan going from in to outside of the cell, whereas gram-negative contain the plasma membrane, the peptidoglycan, the outer membrane, and then many wall-associated proteins, as we can see in association with the outer membrane. So we'll come back to how the gram-positive versus gram-negative bacteria absorb absorb material or metals or ions specifically later when we talk about biosorption. But first, let's talk a little bit more about what can be excreted from bacteria and actually be the outermost surface of the bacterial cell once it excretes this material. For example, we have EPS. EPS just stands for extracellular polymeric substances. The EPS that a lot of bacteria secrete is highly hydrated and therefore functions to absorb material. And we'll talk about what specifically it absorbs or adsorbs later, but basically this function absorption for this EPS material can act to protect cells in what's called microenvironments. And I'll have a video later on talking solely about microenvironments, but basically this is because, as we can see, the EPS can encapsulate bacterial communities or even individual organisms and protect them in a way that maybe their external environmental conditions aren't super favorable, whether it be solution pH, temperature, etc. They can kind of make their own little community environment or just singular environment with their EPS excretions. They can also use EPS to adhere to surfaces to maintain stability. They can also store carbon and energy by excreting EPS and living within it. And they can also bind metals and form minerals using their EPS. And this can be useful for one of two things, typically either nutrient gain, meaning they excrete something that grabs or binds a metal and they take it back up into their cell because they need to use it for metabolism or for growth in some way, or they excrete things to bind to metals that are toxic to them and they want to detoxify their external environment by excreting these things that are going to bind those metals and keep them immobilized. And we'll talk about that later as well. So those are just the general functions of EPS and basically what it is. Now there's also bacterial sheaths, and these are formed in filamentous bacteria or filament shaped bacteria. So long stringy bacteria typically can be encased in what's called a sheath or an outermost surface layer that can either be one of two types. It can be highly ordered protein sheaths or it can be fibular sugar sheaths. And typically all filamentous bacterial sheaths are associated in some way with iron and manganese depositing bacteria. And we've also found some in cyanobacteria. And we've also observed that these sheaths have relatively low permeabilities and therefore might be helpful in terms of protecting against harmful macromolecules. So they're only going to let really tiny molecules through or specific molecules through, kind of like cell walls do, but it's an extra protective layer against harmful macromolecules that may enter the cell had it not had that sheath. And these sheaths also have different surface charge compared to the underlying cell wall of the bacterium. And this may mediate reactions between the cell and ions or solids that are in the external environment. So more mediation or protection against maybe harmful reactions, harmful macromolecules entering the cell, all of that by having this protective sheath layer. And we'll move on to S layers now, but remember, we'll talk about how sheaths might absorb material and what material they might be able to absorb later on in the biosorption section. So now about S layers. S layers are called S layers because they're surface layers. And as we can see, this is a typical bacteria that, from what we can tell, this is the peptidoglycan, so it must be gram positive. Good. And it's got this S layer on the outermost part of it. And these S layers are typically more structured and organized than EPS and sheaths. Because of this, we call them paracrystalline. And they consist mainly of proteins, so they're a proteinaceous layer, as mentioned here, but they also contain the occasional carbohydrate. And they're found in almost all groups of bacteria and archaea, which is the other group of prokaryote we'll talk about in a second. And they're similar in function to sheaths, so they can form aqueous channels that can allow essential nutrients to enter and waste to exit. So they're that extra mediating and protecting layer. And they can also have different surface charge than the underlying cell wall layers. And they can also allow metal binding and mineral nucleation that they can even rid themselves of if the mineralized S layer becomes too heavy or too much of a burden, if too many minerals are nucleating there. And so that's another way to kind of protect or detoxify their external environment, forming these S layers and then allowing mineral nucleation or metal binding and then ridding themselves or shedding themselves of that S layer when necessary. So we'll talk more about what they can bind later in the biosorption section. Now let's move on to archaeal cell walls. walls. Archaea have more variable cell wall structures than bacteria. As we can see, we have the two main bacterial cell wall types shown down here again, and this is gram-positive with the large peptidoglycan layer and the cytoplasmic membrane, and then we have the gram-negative with the inner membrane, the outer membrane, and the peptidoglycan in between in the periplasm, which I didn't mention the periplasm before, but basically it's this part of the gram-negative bacteria that is in between the two membranes, and within the periplasm lies the peptidoglycan of gram-negative bacteria. In archaeal cell walls, they can have just a plasma membrane and an S layer, keeping it simple. Some don't even have the S layer, even more simple. And some have material similar to peptidoglycan called pseudomurin, and some have different structures altogether. And so some of these variations are shown here. But another thing I want to mention regarding archaea is that their phospholipid membranes differ from that of bacteria. I didn't mention what the plasma membrane is made of earlier when we talked about bacterial cells, but basically it's a phospholipid bilayer. And I'm sure if you've taken a biology class, you've heard of this, but what does that mean? Well, it's basically what it sounds like. You've got a phosphol, or the phosphate group, which is on the head side of the phospholipid, and then you've got them connected by ester linkages in the case of bacteria and eukarya. However, they're connected by ether linkages in archaea, and then you've got the lipid, this pink part. That's what the lipid comes from in the phospholipid name. And this lipid side chain can be branched, such as in archaea, or unbranched, such as in bacteria and eukaryotes. So the ether linkage and the branching of the side chain in the phospholipids of archaea is what's different than that of bacteria. Now, there's one more way in which they can differ from bacteria and eukaryotes, and that is that they form a phospholipid monolayer rather than a bilayer. And basically, the bilayer in bacteria and eukaryotes is shown down here to the bottom left of the figure, and you can see there's two phospholipids facing each other, and they form these long chains of two phospholipids facing each other in the plasma membrane. However, in archaea, they can form monolayers of their cell membrane or not, depending on what archaea you're looking at. They're very versatile, and most of the ones that do form monolayer membranes are extremophiles, such as thermophiles or hypothermophiles that love heat. These archaea or extremophiles that live in really hot environments that hardly any other life could handle have an altered membrane in order to allow them to survive that type of environment. And now I'll talk about a lot of other types of extremophiles in my extremophile video, which I will have coming out later on, or if it's out now, go check it out. But that's basically how archaea cell walls differ from bacteria. But what about eukaryotes? So eukaryotes, like I mentioned, include major groups such as plants and animals, as we can see with their cell walls shown here. And you've probably learned from your typical intro bio class that plants have cell walls and animal cells don't. Well, that's kind of true. Animal cells basically just have a plasma membrane, whereas cell walls do have a lot more structure to their walls, similar to prokaryotes. But in this video, we're talking about biosorption, in which we tend to focus on unicellular or microbial organisms, because sorption is all about surface area to volume ratios, and surface area is important for adsorption, as you can imagine. And because plants and animals tend to form multicellular life, the surface area to volume ratio is a bit smaller than what we can achieve with individual cells of bacterial communities or microbial communities, such as algal and fungal communities. And algal and fungal cells actually do a really good job at biosorption. So let's talk a little bit about their cell walls. Algal cell walls have a skeletal layer of inner cell wall fibular skeleton, shown here, with cellulose fibers in a matrix of alginate and fusoidin. I hope I'm saying that right. And within that, they have their plasma membrane, and the outermost layer is an amorphous embedding matrix layer. And because this outer layer consists of this alginate-rich matrix, it consists of a lot of material that is good at biosorption, because alginate consists of repeating units of carboxyl-rich uronic acids, which, as we'll see later, can be really important for the sequestration or binding of certain metals and other cations. And the fungal cell walls also contain a skeletal chitinous layer and then an amorphous outer layer of chitosan, which is just deacetylated chitin. And we'll talk a lot more later about how these amorphous outer layers can absorb certain ions and what ions they like to absorb. So now getting into the biosorption section. How does it work? What is the main driving force behind biosorption onto cellular surfaces? Well, for passive biosorption, and we'll talk later about what active is, for passive biosorption, it has to be their surface charge. That is probably the main driving force behind biosorption. This just means that their cell surface has a charge that attracts the opposite charge. So what is this charge and why does it happen? Well, many organic functional groups that are present at the cell's surface inside the cell wall and meeting with the external environment are amphoteric, and this means that they can either bind or release protons depending on the pH of the solution that they're in. Ionization of these functional groups, which just means the binding or releasing of these hydrogen ions, that's ionization because when they release or bind protons, they're changing their charge and they're going to become either plus or minus some number depending on how many protons they release or bind to. And when this ionization of these functional groups happens at the cell wall, it provides the cell with an electric charge. And this creates an electric field around the cell as shown in this figure. Now, the hydrogen ion exchange is not the only thing that can control its charge. Adsorption of ions can then modify the surface charge. So the whole reason that the ions are absorbing to the cell surface is because of the initial charge due to hydrogen ion exchange. However, if there are ions around, especially like metal or nanoparticle cations, as shown in this figure to the right, we see that the positive cationic charge of these particles is causing them to be attracted to the cell's surface. So what about passive versus active adsorption? So like I already mentioned, this is passive adsorption because the cell's inherent charge is causing an absorption of cations. However, there are more active processes in which the bacteria can employ to go and absorb things. But let's talk a little bit more about why passive metal absorption happens before we move on to the active processes. So experiments have tested the metal binding power of various functional groups at bacterial surfaces, and it seems that carboxyl groups, such as what I mentioned earlier in the outer layer of algal cells, are also associated in the peptidoglycan of bacterial cells. And as the peptidoglycan is the outermost layer in gram-positive cells, as shown here in this figure that we showed earlier, we can see that these cells would be pretty efficient at metal binding, seeing that carboxyl groups have been shown to be the most efficient cationic binding sites, especially for many gram-positive bacteria. But the structure of the peptidoglycan also can impact the degree of metal binding that can occur, because carboxyl and phosphate groups that like to bind cations need to be positioned facing the external aqueous environment. So the positioning of chemical molecules really matters for the reactions that will occur. You can't just bump something into another thing. If it's not facing the right direction, there will not be a reaction, especially if something is bound in a cell wall. It has to be facing the right direction because it can't be moving around freely if it's immobilized in that cell wall. What about gram-negative bacteria? Gram-negative bacteria do not absorb as much metal as their gram-positive counterparts. Carboxyl groups are present in the LPS, or lipopolysaccharides, that are present in the outermost membrane of the gram-negative bacteria. However, only 1/3 of these carboxyl groups are available for binding metals in the external environment because the rest are cross-linked to cationic amino groups. But we talked a lot about how bacteria tend to excrete material like EPS, sheaths, and S layers. So how do these excreted materials help bacteria or hurt bacteria's ability to bind metals? Well, gram-positive and gram-negative bacteria both produce EPS, which, due to its hydrated nature, like we talked about earlier, allows dissolved metals to freely diffuse through the EPS, binding to anionic functional groups along their way. And because of their large reactive surface area, remember surface area to volume ratio is important for absorption, because of this large reactive surface area, the encapsulated bacteria, like we see here, so they're in capsules of excreted material, can bind more metals. Encapsulated bacteria can also tolerate higher metal concentrations due to their higher binding capacities. So if there's too many metals, especially toxic or unwanted metals in the environment, they can handle it because their EPS or excreted material that they are encapsulated within can immobilize a lot of those metals, and the bacteria lives happily ever after in its little microenvironment that doesn't let those metals move around freely and enter their cell and harm them. In fact, the role of capsular production has been thought to be linked to protection against metal toxicity. So maybe it evolved purely for this reason. Anyhow, irrespective of the bacteria, gram-positive or gram-negative, a two-step mechanism has been proposed for passive metal adsorption. Step 1, an electrostatic interaction between the cationic metal and the anionic organic ligand occurs, and step 2, the interaction site acts as a nucleation site for the deposition of more metals from solution and eventually biomineralization, which we will be discussing in the upcoming biomineralization video. But what about eukaryotes? We talked a little bit about how algal and fungal cell walls can be efficient biosorbers. So how does this work? Well, it's similar to prokaryotes. Algae and fungi also have functional groups in their cell walls that can sequester a wide range of metals. In fact, the amount of metal binding in algae increases with metal availability, aka if there's more metals, they will continue to bind more of them. And this can become really important for bioremediation and biorecovery purposes. So if metals are toxic in the environment, we can remediate those environments. We can take out those toxic metals by letting the biosorption capacity of algae help us out with that. Or we can recover economic metals if we can employ the algae in a way where we can use its binding power. And also fungi can bind significant amounts of anionic metals due to protonated amino groups in their chitin and chitosan. So we talked about how they have their skeletal chitin layer and their amorphous chitosan layer. Well, they can actually bind anionic metals because of this. And we talked about how typically cell surfaces have a negative electric field and therefore attract cationic metals, but fungi can actually also attract anionic metals, which isn't unheard of among prokaryotes, but it is not as common as cationic absorption. Finally, we're on to active metal adsorption. So how can microbes actively adsorb metals? Well, first we'll start with the why. Sometimes microbes need metals that are in their external environment for their growth and metabolism, and sometimes they just want to immobilize metals in their external environment so they do not enter their cell and they can protect themselves from toxic metals. And on to the how, in terms of taking up metals for growth and metabolism, they can excrete metal-specific molecules or enzymes that can grab their metal and carry it back into the cell to function in the cell for what they need it to function as. Or if they're trying to immobilize toxic metals and detoxify their environment, they can excrete a similar molecule that's specific to that metal or enzyme specific to binding that metal and then immobilize it in the external environment and not bring it back. And so that's the difference there. In either case, they're excreting organic molecules specific to binding a specific metal. So you can see how amazing biology is that it ever even evolved this function. I mean, it's like, I'm going to put out this thing that's going to grab this exact metal or this exact molecule, whatever it may be, so that I can either take it and use it or, leave it outside of me so I don't have to deal with it. This is just an amazing feat of biology. And possibly one of the best examples is siderophores. Siderophores are molecules specific to binding iron in the environment and then taking it back and using it in their cells, because we all need iron. Humans, any organisms, iron is so important for us. So what we can see is that these siderophores are just molecules that have specific sites with a bunch of oxygens, as we can see, and these sites are where they will grab that iron, and once they grab it in that oxygen trap, they will take it back and use it in their cell. And that is all I have for you guys today. I hope you think this is as cool as I do. I know it's kind of a more biological video than Geo, and my name is Geo Girl, so you're probably wondering why, but I want to come out with more GeoBio videos. One, because I study biogeochemistry, and two, because I really want to talk on this channel more about astrobiology and marine biogeochemistry, which is what I research. And to get to that, I want to lay some groundwork regarding geobiology and biology in general so that we can get into that intersection between geology, chemistry, biology, and astronomy. Also, there are really important topics like bioremediation and biorecovery, which is one of the upcoming videos listed here, and biomineralization, which is obviously includes, some of the things we've been talking about in terms of mineralogy, and also with that bio in front of it. So we'll talk about what those mean in the upcoming videos that we see here, and then later we'll get to maybe some more interesting topics for you. Or if this is sufficiently interesting, I'm glad you enjoyed it, and thanks for watching. I'll see you guys next time. Bye. Oh, and I forgot to mention that the book that I'm using is called Geomicrobiology, or Introduction to Geomicrobiology, by Kurt Konhauser. And I love this book, and it's linked in my description if you want to go check it out. And I'm going to start mentioning which books I'm using for my lectures more often as I move forward with my videos, because I think they can be really useful for you guys. And if you like what I share on my channel, it's all in these things. It's not all from my head, unfortunately. All right, that's it. Bye. Say hi, Ollie. Ollie, look. Oh, there you go. There you go. Good job.